Good morning, everyone. Today we are going to discuss the chapter 3, Human Reproduction. In this chapter, mainly we are dealing with the important points that are gametogenesis, fertilization, zygote formation, blastocyst formation, implantation, gestation, and parturition. In this module, we are mainly concentrating on the important parts of male reproductive system. So here the figure it is given. In this, the important part, that is a pair of testes and the duct system, the glands which are associated, and the external genitalia, or we can say copulatory organ, which is penis. And the main important or the primary sex organ in a human male, it is a pair of testes. And the testis is located within the abdominal cavity in the fetal condition. And just before the childbirth or soon after the parturition, the testis will descend down through a canal. That canal is called as inguinal canal. And it will be well placed within a loose pouch. That pouch is called as scrotum. And why it is located within the abdominal pouch, or from the abdominal pouch it is placed within the or outside the body means, or outside the human body means, within the testis only the whole set of sperms are produced. So the production of sperms is called as spermatogenesis. So the entire process of spermatogenesis is taking place within the testis, which consists of the units of testis, we can say the seminiferous tubule. So within that seminiferous tubule, within the testis, it is produced. And for the proper production of sperms, active sperms, they require 2 to 2.5 degree less than the body temperature. So for that only, the testis is descending down from abdominal cavity to outside the body. And when the testis will start functioning, when we are asking, it is actually from the pubertal age onwards. So in human male, the puberty starts by the age of 12 to 14 or even 15 years of age. And from that time onwards, it is a continuous process. Continuous process means till the death, the sperm production will take place. And that too, each ejaculation, or means the release of sperm, that it will consist of approximately 3 to 4 ml. So that semen consists of, each ejaculation consists of nearly 300 millions of sperms. So the number of gamete production in human female is millions. Compared to female, that is millions in number. So it is produced within the testis. So this testis is placed outside. That is, which require that whole production required to 2.5 degree less than the body. Then not only that one, according to the body environmental conditions, the scrotal muscles will either contract or relax. If it is summer season means it will relax and the testis will be placed slightly away from the body. And if it is winter means the scrotal muscles will contract towards, because outside it is temperature is very less and inside the temperature is more. So the testis will be, when the contraction of scrotal muscles, that will place the testis slightly towards closer to. So in that way, always it will ensure that the temperature is maintained, which is actually always less than 2 to 2.5 less than the body temperature. And when we are moving into more in detail, the testis consists of, or we can say it is well placed in scrotum, and it requires less number of temperature. And the size of the testis, when we are saying it is oval in shape, and it is 4 to 5 centimeter long and 2 to 3 centimeter wide. And inside the testis, when we are saying, it is consists of nearly around 250 lobes. Each testis consists of 250 lobes. And this lobes consists of, this is the figure, each testis, the LS of testis we can say. And inside the testis also a set of accessory ducts are there. And even outside the testis also a set of accessory ducts will be there. So that we can divide into intratesticular duct system and as well as the extratesticular duct system. So here, when you are observing the duct system, that is intratesticular duct system, and you see inside the testis there are many lobes. You can see here many lobes inside. So nearly 250 lobes, and in each lobe here, diagrammatically it is represented only one seminiferous tubule. Actually, it can be in each lobe, it can be 1 to 3 seminiferous tubule. It is highly coiled and packed very closely. So in each lobe, this seminiferous tubule as such, when you are saying each tubule is, it is a loop, like it is arranged in the form of a loop, like with two, both the ends will be joined together and goes out of the testicular lobule in the form of a straight tube. So that straight tube region, we can call it as tubuli recti, and it is not of that much of importance. Next duct only we have to say, that is seminiferous tubule, which passes through a straight tube, and then it will interact or interconnect with each other. So all these 750 seminiferous tubules comes out of the testicular lobes, and they will unite together and they will interconnect each other. So that interconnected network of tubular system or duct system is called as recti testis. So seminiferous tubule, which passes through straight tubular region, that is called as tubuli recti. Then they will interconnect each other in the form of a network. So that is why the term reticulum, that is the word meaning, reticulated tubular system. So that is why reticulated duct system, it is called as recti testis. Then recti testis will join together and they will emerge out in the form of little more straight tubes. That region, it is somewhat, it is called as vas efferentia. Vas efferentia. So the number of vas efferentia, they will join together and they will again divide into nearly approximately 1 to 20 tubes are there. So that tubes are called as vas efferentia. So seminiferous tubule, then recti testis, then vas efferentia. Okay, these duct systems are enclosed within the testis, hence it is called as intratesticular duct system. Then this vas differential joined with a long, highly coiled tube, which is actually overlapping the region of the testis. So it is highly coiled, and that region, from that region onwards, it is categorized as extratesticular duct system. So extratesticular duct system starts with epididymis, which is highly coiled. And there only, so continuously within the seminiferous tubules, the sperms will be produced that will pass us through the tubuli recti, then recti testis, then into vas deferens. Then it will be passed through the epididymis, and within the epididymis, it is temporarily stored. So the purpose of epididymis is the sperms are temporarily stored within the epididymis. And then the sperms will pass through highly coiled, pass through a tube, that tube that is called as vas deferens, which will overlap. The vas deferens will overlap. Here is the, in the figure you can see very clearly, the vas deferens, this is the region, the vas deferens, which will overlap the urinary bladder, which will bend over the urinary bladder. So the sperms will pass through the vas deferens when a person is sexually excited. So when the person is sexually excited, which is the sperms which are stored already within the epididymis will carried away, because the lining of the vas deferens is ciliated. So by the ciliary movements of the lining of the inner lining of the vas deferens, the sperms will be gradually, it is driven and it will be passed over through this. So this is also you can call it as extra testicular duct system. So meanwhile, it will be collecting, so that only here we said the duct system vas deferens, and we'll move on to. So the vas deferens will bend over the urinary bladder, and there at the junction of that urinary bladder, it will receive the secretions from 3 accessory glands. So the first gland, we can call it as the seminal vesicle, which is highly enlarged and it is leaf shaped, and they contribute the more fluid secretion. Approximately 70% of the fluid secretion will be contributed from the seminal vesicles, and it mainly consists of fructose, calcium, inositol, prostaglandin, and certain enzymes. And so 70% of the total semen or the fluid part of the, or we can say the total secretion, it is contributed by the seminal vesicle. And then that duct will pass through another unpaired glandular region. That gland is called as prostate gland. And the prostate gland also will contribute approximately 20 to 30%. It can be up to 30% of the fluid part, which is slightly milky in appearance, and it is alkaline also, by milky and alkaline secretion, which will neutralize the acidic hyaluronic acid which is present within the genital tract of the female. So it can neutralize the acidity of the genital tract and can drive the sperms through the easy movement of the sperms through the genital tract when it is inseminated. So it is prostate gland, it is milky secretion, and that milky secretion also will produce certain characteristic smell or contribute certain characteristic smell to the semen. Then again, it will collect a small secretion from one more gland. It is very small and peanut shape only, and that is called as Cowper's glands or bulbourethral glands. So all these three glandular secretion is actually activate, or we can say it will contribute plus sperms together, we can call it as semen. So this is the main role of seminal plasma. The fluid secretion of these three glands together, we can call it as seminal plasma, which consists of the secretion of seminal vesicle, prostate gland, and Cowper's glands. So these ducts, so when it collects these secretions, that duct which is vas deferens will continue as a duct. That duct is called as ejaculatory duct. Then at the base of that one, the Cowper's glands, soon after the Cowper's glands, the duct will continue and it will pass through the copulatory organ, which is penis, through the middle region or to the center of the penis, and that time the duct is named as urethra. Okay, urethra, and this urethra will pass us through the center of the penis, and then it will opens out through an external opening that is called as urethral meatus. So here the semen sperms will be produced within the testis, and from the testis it is passed through the duct system, that is intratesticular duct system, and then it will pass through the extra testicular duct system, that is vas deferens, then ejaculatory duct and urethra. And when it is reaching the ejaculatory duct, it will collect the secretions from the secretions from seminal vesicle, bulbourethral gland, and then prostate gland, and it will continue as a urethral duct which passes through or penetrates through the penis, and which is opens out through an opening that is called as pineal urethra. So the discharge of the semen, when we say semen, it is actually seminal plasma plus sperms. Okay, and each that release of semen from the penis, it is called as ejaculation. And there is a small difference between ejaculation and insemination. Ejaculation means it is the release of semen outside the body. Release of semen outside the body, it is called as ejaculation. The same way, the release of semen into the vaginal tract of the female, then it is called as insemination. Into the feminine tract, it is called as insemination. So ejaculation and insemination is different. So when it is ejaculated or inseminated, normally in a normal healthy person, the total amount of semen will be 3 to 4 milliliters per ejaculation, which can contain approximately 300 millions of sperms in a normal healthy person. And if it is a normal count, when we'll say sperm count, that is mainly very important factor in fertility, determining the fertility rate. So minimum of 40 to 40 percentage, or we can say at least 80 to 120 million sperms must be there in a per milliliter of sperm semen. So if it is less than that, sometimes it may lead to more complications and it can lead to infertility conditions and all. So normally in a person, approximately 60% of sperms will be functional, and in that 40 will be movable. The very healthy sperms should be minimum, it must be there when they are producing. So we will move on to more in detail structure of testis. So here in this slide, the testis are just an external characteristics the internal characteristics we are going to discuss. That is, the testis is covered by a dense covering tissue that is called as tunica albuginea, and it consists of nearly 250 lobes. Each lobe there is approximately 1 to 3 seminiferous tubules are there. It is highly coiled and packed within the body. And then the next one it is, so here you can see the next slide, you can see the structure. So each seminiferous tubule itself is lined by, so there can be many coils of seminiferous tubules. So each seminiferous tubules are highly coiled and it is lined by two types of cells, spermatogonia and pyramidal shape of cells, that is called as Sertoli cells. And the spermatogonia only undergo repeated divisions, and finally from each spermatogonia there can be formation of primary spermatocytes by mitotic division, and the primary spermatocytes will undergo first meiotic division and they can form secondary spermatocyte, two secondary spermatocyte, and each secondary spermatocyte will undergo second meiotic division, and then they can form from each secondary spermatocyte two spermatids, and that is haploid. Even secondary spermatocyte also haploid, and altogether from a single primary spermatocyte there can be 4 equal spermatids can be formed. Then there will be a morphological changes, that is spermatid will transform into a tadpole-like structure that is called as spermatozoa. So each primary spermatocyte, when they complete meiosis, each 4 equal size spermatozoa which is functional. So 4 sperms can be produced. So the spermatogonia and primary spermatocyte also diploid, and as well as the secondary spermatocyte is haploid, spermatid also haploid, and spermatozoa also haploid. So we can say the Sertoli cells are nutritive in function. It will provide nutrients for the developing sperms. When the spermatozoa, soon after the transformation, they will get embedded within the Sertoli cells, because they have to grow and mature. So soon after the formation of spermatid to spermatozoa, that stage, the last stage of the spermatogenesis is called as spermiogenesis, because the globular spermatid, which is round in shape, will have to transform into a tadpole shape. So there is a morphological changes there occurs. So that morphological transformation of spermatid to spermatozoa, the last step of the spermatogenesis, is called as spermiogenesis. So often they'll ask the difference between spermatogenesis and spermiogenesis. Spermatogenesis means the entire transformation of spermatogonia to spermatozoa is called as spermatogenesis. It is taking place within the testis. And the spermiogenesis means the last step of spermatogenesis, that is spermatid to spermatozoa. There the morphological changes occurs, and they will transform into a head, neck, middle piece, and a tail. But even then, that spermatozoa is not completely developed. It will get embedded, or it will get attached to the Sertoli cells, and from the Sertoli cells the nutrients will be absorbed. Then it will gradually mature. So the mature spermatozoa, after the complete development, it will detach from the Sertoli cells, and it will be detached into the lumen of the Sertoli cells. That process is called as spermiation. So here in this topic we have discussed with 3 terms, that is spermatogenesis, spermiogenesis, and spermiation. So spermiation is the detachment of matured mature spermatozoa from the Sertoli cells into the lumen of the seminiferous tubule is called as spermiation. Then within the interstitial space, that means the space between the seminiferous tubule, within the testicular lobes, there is a mass of cells. That mass of cells are called as Leydig cells. And the role of Leydig cells is they secrete the male sex hormone. Collectively, we can call it as androgens, and in that, most important androgen is testosterone, which is a male sex hormone. So the main primary sex hormone, or the collective term for the androgen, is testosterone. So here in this figure, you can see the mass of cells, which is called as Leydig cells, which will secrete the androgen, and this androgen testosterone control or regulate the whole process of spermatogenesis as well as the secondary sexual characteristics in human male. That means the muscular behavioral pattern and the muscular nature of the male, then broad chest, and then low pitched voice, and excess hair growth, etc. in our body parts. All these things are regulated by the androgen. Then when we'll move on to the next one, important points of this one. So the figure is very important, that also they will ask for exams and even for the practical also. So all these points we have discussed. Then apart from the Leydig cells will produce testosterone, and apart from that there is one more type of cells will be produced by the Leydig cells, that is called as immunogenetically competent cells. So what do you mean by immunogenetically competent cells? Means that whenever in our body, when we are infected with any pathogens, there is a chance for entering this pathogen, bacteria or virus and all, even to the into our body, into the testis also. So within the testis, if any pathogen is entering, means with the help of immunologically competent cells, normally it is lymphocytes, mast cells, and then macrophage cells only. So if any pathogens are entering, these immunologically competent cells, which is coming under the category of lymphocytes and white blood corpuscles itself, and they will engulf the pathogen and they will protect from the infectious conditions. So the testis also very well protected with the help of immunologically competent cells that is produced by the Leydig cells. Now we'll move on to the graphical presentation of the structure of TS. So the first left side, the figure which is given is in your textbook, it is there. Sometimes the figure as such they will give, and they will ask you to label the parts and explain the role of. So whenever you are, or even sometimes the ploidy of the cells, they will mark it and they will ask you. So in the ploidy, when they are marked, the spermatogonia and the primary spermatocyte is diploid, then secondary spermatocyte, spermatid, and spermatozoa also diploid. So what about the ploidy of Sertoli cells? Sertoli cells, definitely it is diploid. Okay, Sertoli cells is diploid. Then lumen, they will ask you to label the lumen, and the Leydig cells, mass of cells, Leydig cells, and what is its secretion, what is the role of Leydig cells. All these points will be asked you to explain. Now we will say, so from this the sperms will be produced, and after that sperm production, it will passes through the accessory ducts and it will collect the secretions from the seminal vesicles, prostate glands, and the Cowper's glands. Then that secretion together, so that time we'll call it as semen. Semen will pass through the copulatory organ. So the main copulatory, or the copulatory organ, or which consists of external genitalia, is called as penis. And the penis consists of three rod-like structures. Inside the penis, it is three rod-like structures are called, which is spongy in nature, that is, and it is erectile. It is made up of erectile tissue, and that erectile tissue is filled with the air spaces. So when it is sexually excited, the blood will rush into that erectile tissue, that will make the penis more erect, and the tip region of the penis is highly glandular, and that region it is slightly bulged and swollen, and that region it is called as glans penis, and it is most excitatory region, and it is more glands will be secreted, and they secrete more lubricating agents which will help in lubricating the penis at the time of insertion of penis into the vaginal tract, or we can say copulation. And this glans penis is covered by a fold of skin. That fold of skin is called as prepuce or foreskin. And this foreskin, in surgically, to avoid infectious condition, it can be removed, and that procedure is called as circumcision. In some religions, circumcision is a part of their religious customs, especially in Muslims, Jews, and all. The circumcision is a part of their religious custom. So it is medically also it is advantageous, because if you remove that the tip region of the foreskin or the prepuce, it will avoid inflammatory chances due to the viral or bacterial conditions and all. Then we'll move on to the next topic, that is the semen. So we haven't discussed too much because the components inside the semen. So the male accessory glands, which mainly consists of paired seminal vesicles, a prostate, single unpaired prostate gland, and paired bulbourethral glands. Another name of bulbourethral gland is Cowper's glands. They secrete the collection together, we'll call it as seminal plasma. The seminal vesicles, that itself, three types. Seminal vesicles consist of approximately 60 to 75% of the fluid in semen, and it consists of rich in it makes it is rich in proteins and enzymes, mainly fructose, vitamin C, prostaglandin, then phosphoryl, choline, etc. Then the high glucose content provides nutrient energy for the spermatozoa. So nutrient is a carbohydrate. So energy is given by the fructose. Then the second one, it is prostate gland, which is single, and it is secretes a slightly alkaline milky fluid, and this helps in the survival of the sperms in the acidic vaginal environment, because vagina secretes certain hyaluronic acid, we can say. So that acidity will be neutralized by the prostatic secretion, hyaluronidase, we'll say. Then the secretions also improve the motility of the sperms. Then the last gland, it is paired only, that is called as Cowper's glands or bulbourethral glands, which will secrete more mucus secretions, and which will add up or which will help in lubricating the penis and neutralize, and if any acidity is there, means that also will be neutralized. So already I said the semen in per ejaculation, it consists of 3 to 4 ml, and each the whole thing together collectively 200 to 300 millions of sperms, and the sperm count for a normal person, when we will say normal sperm count, will come within the range of 80 to 120 millions of sperms per 1 ml. So if it is less than 40 million per 1 ml, means that person can lead to infertile conditions. Now we will discuss about a summary of spermatogenesis, that is formation of sperms from spermatogonia, which is a diploid cell. It takes place inside the seminiferous tubules of the testis, and the spermatogonial cells in seminiferous tubules undergo repeated mitotic division. And already I said there are two types of cells, type A cell and type B cell. Type A cell will retain their continuous multiplication capacity, and they will act as the stem cells, and type B cell will modify into primary spermatocyte, which is diploid. Then this primary spermatocytes will undergo first meiotic division. Two daughter cells will be formed, that is called a secondary spermatocyte, which is haploid and it is equal in shape. And each secondary spermatocyte again further completes the second meiotic division, and from each secondary spermatocyte, two daughter cells, that two daughter cells are called as spermatids. So since there is two secondary spermatocytes are formed, four spermatids are forming. This spermatids will undergo a transformation or differentiation, you can say. It is morphologically differentiated into a tadpole structure, that is called as spermatozoa. So the last step of spermatogenesis is called as spermiogenesis, and this spermatozoa will get embedded within the Sertoli cells, and there it will absorb maximum nutrients, and they will grow and they will mature. The mature sperm, automatically it will be released from the Sertoli cells. That process is called as spermiation into the lumen. Then through the lumen it will be carried, and through the accessory ducts, everything. Then finally it will be ejaculated out in the form of semen. So the duration for the formation of one sperm from the spermatogonia, the total duration, it takes place around 72 days. So here this is the figure, very important figure. There one side it is given the pattern of meiotic division, mitotic division, then meiotic division, and on the middle region they have represented the how the proliferative stage, or we can say the maturation, multiplication phase, then growth phase, then division phase, etc. And on the right side the ploidy of the cells also are given. So this figure is very, very important. And here, if you want, you can represent the spermatogenesis itself, the diploidy, haploidy, all those conditions with the paired sets of chromosomes. So different patterns of representation is included. And when we move into more detailed structural characteristics of sperm, when we are saying sperm is the male gamete, and millions of sperms will produce from puberty till the death. And the sperm consists of or the main parts of the sperm is, it is a tadpole, it's having a tadpole shape, and the size is approximately 60 micron, and the number already we have mentioned. And then the amount we already, semen I said, and even the presence of, I said, the semen consists of fructose. So it is the only semen only contain the fructose. So mainly, if any body fluids which shows the which shows the presence of fructose, means there can be, that is the indication of ejaculation or insemination. So rape test and all, mainly we are detecting whether in the body secretion or anywhere in the body or in the cloth, if there is fructose is there, means there we can, it can provide or it can give you a clue about the sexual abuse as well as the rape which is taking place. So it is a very good forensic evidence for determining the body secretion, whether it is semen or any other body fluids. So it is also called as detecting the presence of fructose within the body fluid. It is called as rape test also. Then infertile condition, already I said, less than 40 million in 1 ml of sperms, means that can lead to infertility. Now when we move on to this figure, you can see the human sperm. It is having a tadpole shape, and it consists of major 4 parts, that is head, neck, middle piece, and a tail. The head consists of a large nucleus which consists of haploid set of chromosomes. That means as a result of meiosis, the diploid condition is reduced into haploid, so only 23 chromosomes will be there within the sperm. So that 23 sets of chromosomes will be there in the, it will be enclosed within the nucleus, and it is surrounded by a very thin layer of cytoplasm, negligible amount of cytoplasm. And also along with that, there is an oval structure or oval gland, we can say, that is called as acrosome. This acrosome is, which is cap shaped on top of the nucleus only, it is arranged, and it is believed to be the modified Golgi body, which consists of a lot of powerful enzymes. Collectively, that enzymes which is present within the acrosome is called as sperm lysine. And this sperm lysine helps in dissolving the egg membrane of the ovum at the time of fertilization. So the main components of sperm lysine, it is not mentioned in your textbook, but even then, the enzyme that is called as hyaluronidase, then corona penetrating enzyme, because it is the outer line of the ova, the corona radiator, we can say. So the cementing substance between the corona radiator is dissolved by corona penetrating enzyme, and inside that there is again one more thick layer that is called as zona pellucida. So that layer also can be dissolved with the help of this enzyme. So mainly the components of sperm lysine is hyaluronidase, corona penetrating enzyme, and zona lysine. So this acrosome, when the sperm is in contact with the egg membrane, immediately the acrosome will rupture, and they will release these powerful enzymes which will dissolve the membranes of the outer line, and as a result there will be that will give a pathway for the nucleus to enter into the ova. So the nucleus, when as soon as the nucleus is entering into the ova, that process is called as fertilization. There, this set of nucleus and the ova, the nucleus together, they will meet together, that only the fertilization we will say. So half the set of chromosomes are conserved within the nucleus of the sperm. So that is the head. And the next region we can call it as the neck. Neck consists of 2 centrioles, which is called as, the first one is called as proximal centriole. Immediately below that there is another centriole that is called as distal centriole. It helps in the formation of spindle fiber formation at the time of cell division. And then the attachment, there is one more centriole will be there, that is located at the end of middle piece. And so three centrioles are there: proximal centriole, distal centriole, and a ring centriole at the middle piece also. Then middle piece consists of, the neck is only a short segment, then it is followed into middle piece. Middle piece consists of numerous mitochondria, and mitochondria is the source of energy. So the energy for the sperm to swim in the genital tract, that energy for the formation of the movement of the sperm is given by the mitochondria. So each mitochondria is coiled, and there can be approximately 10 to 14 coils of mitochondrial arrangement. And through the center of the middle piece, from the distal centriole, there is a filament arises. That filament is called as axial filament, which will pass us through the tail also. Tail is surrounded by the central axis. Centrally, it is consists of axial filament surrounded by the cytoplasmic sheath. And at the extreme tip, that cytoplasmic sheath is not there. That extreme tip of that tail is called as end piece. So the main important functions: head is providing a chromosome, at the same time the chromosome, sets of chromosomes, and then the middle piece, mitochondria, the energy supplement, and the tail helps in swimming in the, or movement, helps in movement of the sperm within the genital tract. The last topic of this topic is the testicular regulation, or we can say the hormonal regulation in human male. And here the regulation starts from the main organ, that is the master gland, the super grandmaster, that is hypothalamus. So hypothalamus will secrete a set of hormone. In that, the sex hormone, when we are saying, it is called as gonadotropin releasing hormone, or short form is GnRH. So this gonadotropin releasing hormone will be passed through the hepatic portal system. Then it will reach into the anterior pituitary. So when it reaches into the anterior pituitary, it will stimulate the gonadotroph region of the pituitary, and it will stimulate the pituitary to secrete two hormones. So pituitary hormones, that is called as FSH and LH. FSH means follicle stimulating hormone, and LH means luteinizing hormone. So here the FSH and LH from the anterior pituitary, it will directly stimulate the testis. So from the brain, the hormones will be released into the, that means from the pituitary it will be discharged into the blood, and the blood will supply to the testis. So on the testis, there are certain receptors which will absorb, or which will bind with FSH and LH. So FSH will act on Sertoli cells in the testis, in the seminiferous tubule, and this Sertoli cells will stimulate to produce two proteinaceous factors. One is called as androgen binding protein, and another one is called as inhibin. So inhibin will inhibit the spermiogenesis to a certain extent, or spermatogenesis as such, a negative feedback. And ABP means androgen binding protein, that helps in retaining androgen, that means testosterone, within the lumen of seminiferous tubule. So always within the seminiferous tubule, they will ensure that because of this protein, it will bind with the testosterone, and the viscosity will be more, so it will retain within the seminiferous tubule. So this androgen itself, testosterone itself, they will stimulate the spermatogenesis. That means the formation of sperms from spermatogonia into primary spermatocytes, secondary spermatocytes, spermatid, then into spermatozoa. Then that spermatozoa only will bind with the seminiferous tubule. There the spermiogenesis taking place. So Sertoli cells will directly influence the spermiogenesis, whereas LH means Leydig cells, which is produced by the interstitial cells. And that LH will stimulate the interstitial cells, that mass of cells between the interstitium of the seminiferous, within the testis, that will secrete the testosterone, which is a primary hormone. And the testosterone will regulate the entire process of spermatogenesis, as well as the secondary sexual characters. Okay, so secondary sexual characters are mainly exhibited in the presence of testosterone. So in the early childhood, the testosterone is less in amount, means that can also lead to from feminine characters exhibited by the person. So the last slide of this session, it is the graphical representation. So you have to make note. That is the hypothalamus. Hypothalamus will produce GnRH. It will act on anterior pituitary. In the anterior pituitary, again they will be stimulated. They will secrete two pituitary hormones, that is LH and FSH. FSH will stimulate, will act on both the hormones will stimulate the testis, and the separate cells, that is LH will act on Leydig cells, whereas FSH will act on Sertoli cells. So Sertoli cells will regulate spermiogenesis through ABP. ABP means androgen binding protein, as well as there is a kind of inhibitory activity, that is by the help of inhibin. So when these activities are more, it will be suppressed by a proteinaceous factor, that is called as inhibin, which will inhibit the, or which will act on hypothalamus. So the GnRH will be less, and anterior pituitary also it will act. So as a result of FSH and LH. So in that way there is a regulation. Okay, so there is an inhibitory regulation will be there. Whereas on the other side, LH will stimulate the testis, the Leydig cells, and as a result, testosterone will be produced. Testosterone always will stimulate the spermatogenesis. Be very careful. Here it is spermatogenesis, here it is spermiogenesis. So here it will stimulate the spermatogenesis. Spermatogenesis again, it will act as a positive control only, that is it will stimulate, it's actually stimulate, it will stimulate more and more production of spermatogenesis, whereas inhibin excess amount of testosterone. Once again, if too much of testosterone is produced than the normal, then it will show an inhibitory effect. So inhibin also will show inhibitory effect. Testosterone, excess amount of testosterone also will show inhibitory effect. Whereas in normal amount of testosterone means it will show always stimulatory effect, and it will regulate the spermatogenesis. So with this, we have come into a conclusion of the major topics. Just a recap of this topics we have discussed. It is male reproductive system, that again consists of the main sex organ, that is a pair of testes, which is located within the scrotum. Then accessory ducts such as seminiferous tubule is the main tubule. Then from there retitestis, vas efferentia, then epididymis, vas deferens, then ejaculatory duct, then urethra. And urethra opens out through urethral meatus. That opening is called as urethral meatus. Then along with that, male's accessory glands also, which is opened into the ejaculatory duct, seminal vesicle, prostate gland, and bulbourethral gland. Collectively, we'll call it as seminal plasma, which will give, which will add up more nutrients as well as many enzymes which are required for the proper formation of the proper activation of the sperms and the lubrication of the penis, etc. Then within the testis, again it consists of testicular lobes. It consists of highly coiled tubes. The tubes are called as seminiferous tubule. Each tubule is made up of two types of cells, that is spermatogonia and Sertoli cells. Sertoli cells are nutritive in function. Spermatogonia transforms into spermatozoa by spermatogenesis, and between the seminiferous tubule there are mass of cells, that is called as Leydig cells, which secrete the nutrient, which will secrete the testosterone. It is the main male reproductive structure which will regulate the entire process of spermatogenesis. So with this, we have come into a conclusion, and here the next module we will discuss about the male reproductive system. Thank you very much.